I'll start off by having the three panelists briefly introduce themselves and speak a little bit about their background and the company that they're currently working on. I'll start with you, Dan.
Thank you, Stefan. I'm Dan Elbaum. I am the Chief Scientific Officer at QurAlis. We are a precision neuro company focused at the moment on ALS. We also have a PSP program as well as a Fragile X program. The ALS programs are actually in the clinic, and the other programs are preclinical. I'm probably the token chemist on the panel, talking about the diversity of scientific thought. I'm a chemist by training. I spent many years of my life as a medicinal chemist, and now I do pretty much everything up until you get to a human being. So it's nice to be here.
Beth, over to you.
Thanks, Beth Hoffman. I'm founder and CEO of Origami Therapeutics. My background, I've been in industry at Lilly, Amgen, and Vertex, not here, but in San Diego. So I've had quite a bit of experience putting mainly small molecules into the clinic. So basically, I started Origami Therapeutics to sort of riff or leverage on what I learned working on the cystic fibrosis drugs. These drugs out of Vertex have really transformed the way cystic fibrosis patients are treated and the way they look at their lives, and the hopes and dreams actually come alive. And so with my background in molecular and cell biology, as well as mainly in the area of neuroscience, I saw some similarities, mostly around protein folding, that made me think that we could apply a lot of what we did in cystic fibrosis to neurodegeneration. And so I left Vertex and started Origami Therapeutics, and our first indication is Huntington's disease. And really the reason for that is, like Daniel said, you know, we're focused on, you know, the neuro form of precision medicine, and that is looking initially at monogenic diseases. And so our lead indication is Huntington's disease, and we have a follow-up program on tau, frontotemporal dementia. And so I'll hand it over to Christian.
Hi, I'm Christian Marescu, Senior Vice President of Biology at Vigil Neuroscience. I lead everything from as folks know in biotech, you wear many, many, many hats, and you guys know this. Everything from, you know, ideation on early pipeline projects to supporting the biomarkers for, you know, our 2 or 3 clinical programs. So our efforts in the clinical space are focused on 2 indications in principle. One is a rare disease, which I was so excited to hear somebody mention at this event. It's called ALSP, adult onset leukodystrophy with axonal spheroids and pigmented glia. You get very good at saying these things. It's not ALS, but it's a microgliopathy, and so it's quite rare. And I also work on a program in phase 1, which is for Alzheimer's disease. All of these efforts are focused on, you know, modulating microglia, inspired by some of the human genetic insights that Dr. Sai and Dr. Selko referenced before. So, pleasure to meet everybody.
Great, well, thank you. So we'll dive into some questions I have for the panelists, and of course we'll leave some time for questions at the end from the audience as well. We'll start with you, Beth. At Origami, your company is focused on clearing toxic proteins, which we've heard a lot about today, and implied for the mechanism of action in many neurodegenerative diseases. So can you tell us a little bit how you're using this technology to combat Alzheimer's and other neurodegenerative diseases, and how you leverage that on the preclinical side through phenotypic screenings as well?
Sure, thanks so much for the question. You know, so I started with the end in mind. So what is it that we need to see happen in patients? What are we looking to try to do? And you work backwards. And then the other two, basic principle for me is, that we have to be able to do the clinical trials, So what do you need for that? And then to think about, what does that mean we need to do to try to find the drugs? And so you've already heard from Dr. Tsai about phenotypic screening. We took a phenotypic approach, but not, such a broad phenotypic approach. We were looking for what we wanted to see in patients. In our case, in Huntington's disease, a lowering of mutant Huntington. And we basically said, what's going to be the home run? So I don't want to do a little. I don't want to pretend that, it's okay if we get rid of the normal Huntington. I want to restore normal function, or even be able to prevent disease. Because as we've heard, the earlier you can get in before symptoms appear, and more importantly, before brain cells die, this is really the direction we need to look at. And so that's something you can do if you're going after a genetic form of the disease. And so basically that's what we did, is we did phenotypic screen. And I think, a lot of times people hear that and they go, oh my God, who's going to fund that? Well, that is a good question. But how are we going to do that, and how are we going to show target engagement? And I think that there have been some huge advances in proteomics, in trying to model protein folding, and that you can very systematically work through how do you think your protein's working, and now figure out what the target is. And so I think that the rationale for phenotypic screening, I think, is raised to a new level. I think I'll just add, there are lots and lots of genes. We haven't really been that good at picking out what the right genes are to treat a disease. And I say this because I've been party to that at a number of very fine pharma institutions. And so I think for me, it was important to really go after what we needed to, focus completely on the patients and what we knew caused the disease.
Thank you. Christian, you've been at Vigil Therapeutics, focused on microglia as a therapeutic target. Can you tell us a little bit more how you're using that platform to target neurodegeneration and neuroinflammation in particular?
Yeah, sure. Thanks. So I think there's a very interesting feature of how we go about often in the past thinking about interrogating our targets. And you mentioned phenotypic screens, and maybe there's even some intermediate, which is just making sure the proteome in your cell system is relevant. And a lot of the causal or the risk-associated genes for, say, Alzheimer's disease, which we heard talked about, have very, very poor mouse orthologs. Mm-hmm. So, and probably this makes sense, right? Evolutionarily, mice are running around and exposed to very different environments. Their surface proteomes are going to be tuned to a completely different environment than our own. And but this creates a challenge. And so as a platform, we have really emphasized being able to screen and understand biology and relevant systems, to one, model a disease state such as ALSP, which is very rare. You can't get access to a lot of patient samples. This is a challenge in the rare disease space. But there's also no relevant animal model, partly, that captures these full cardinal features that is represented in the patient population. And so we really rely on very fine molecular understanding and mapping. And for all that, you really need a human system to understand what protein interacts with the other protein, and how does that all map together in a network, a causal network, whether it's Huntington's or CSF1R, which is the gene, causal gene. So I think, again, we're trying to treat human diseases, and it makes, therefore, simple sense work in an appropriate model system, even though that can create challenges, and you have to solve those challenges in order to just raise the bar for yourself. Do you sacrifice a little bit on throughput? Does it cost a little bit more money? Sure. But are you getting to a better molecule or a better understanding of what kind of pharmacology you are really having? I think that trade-off is well worth it. And that's where I think, working in human systems is really imperative, given that, at this stage, there's kind of not much of an excuse to work in any other system otherwise. Yeah.
Great. All right, so we've heard now about targeting microglia, targeting toxic proteins. I'm going to hand it over to Dan to speak about QurAlis' antisense oligonucleotide platforms as a tool to modulate splicing, and how you guys are using that to treat ALS and for other indications.
Thanks, Stefan. So for those of you who don't spend every waking moment of your lives thinking about oligonucleotides, there are basically two flavors of ASOs. There are the gapmers, the ASOs that knock proteins down. They engage RNase H. QurAlis for the treatment of ALS to knock down SOD1 is sort of the most famous one at the moment, although there are others, certainly in the periphery. The paradigm that we're looking at is much more geared towards splicing and correcting missplicing that occurs in disease systems. And so in this case, you're actually not engaging RNase H. You're trying to correct a splicing event that has gone awry. The most famous of these is Spinraza for the treatment of SMA. And we're focused, at least in ALS, which I can talk about, on two targets. One of them is Stathmin-2, and the other one is UNC13A. And so, we have looked at various splicing events that have been shown to occur in ALS patients and looked to correct those. And we are largely focused on human systems, on iPSC-derived motor neurons from patients or from wild type individuals, normal, healthy individuals. To examine these splicing events. The human systems are actually very important here. As Christian mentioned, mice actually don't get ALS. And in fact, if you induce some of the pathologies in mouse cells, you do not see the same missplicing events that you see in humans. And so using those animals for studying these phenomena is pretty much useless. So, there are a lot of splicing events, particularly if you look in the CNS, a lot of very large introns. And so we're certainly bullish and of the opinion that there will be a lot more targets that will come out in the CNS that are ameliorated by correcting their splicing. So.
Great. So I think both Dan and Christian have touched on the need to look at human systems for a variety of reasons, let alone that mice typically don't get even old enough to develop neurodegeneration. So all three companies, you're using a sort of keyhole indication through some genetic group of patients to advance your drugs. And what do we see today on the preclinical models that help us to potentially de-risk some of those activities before we're in the clinic? So what are you in your company, or what do you see other companies doing in terms of advancing those models, be it humanized mouse models or organoids or iPSC technologies? Maybe we'll start with Christian there.
Yeah, so preclinical models often comes up as a gap area, right, in our field, and how do we utilize them? I think, I mentioned this rare disease, ALSP, and I mentioned there's no preclinical, or at least mouse model of this disease. So what can we use in those systems? Well, I referred to this in my responses. You have to really understand and come up with a very refined mechanistic hypothesis. And in those settings, it's best, where the models say are not translational, it's best to stay close to the proximal causal biology of the disease. And so you stick close to what matters in the patient. So whether that be a multicellular, organoid or triculture system or some mouse model, if that works in that scenario, I think you're advised to, ask what you want to ask of that mouse model system. If you think it's going to tell you about efficacy, well, good luck. I hope you're more fortunate than the most of us, but might it be helpful? Let's take the case of, say, Mouseheimer's models, right? Can that tell you maybe a little bit of how the biology of your novel target works and what types of readouts you may expect to see is part of your translation in the clinic? Sure. I think you can utilize those preclinical systems to maybe forecast what your plans will be. But I think as efficacy readouts, these systems are, they are what they are. They're very challenging and limiting. Yeah.
Beth, do you want to?
I think we all agree that you need to look at human systems in large measure because a lot of what we're talking about are protein-protein interactions. And so there are multiple examples out there where a single amino acid difference actually completely changes the pharmacology. I can cite you the example of serotonin 1B versus 1D receptor. Everybody thought these are going to be completely different genes. They differ between 1B in mouse and 1D in human by a single amino acid. So I think the importance of using the human systems can't be overemphasized. That said, we're kind of fortunate in Huntington's disease in that actually the models are not half bad, but it depends what question you're asking and what you want that model to tell you. Is it going to look like the human disease? No, there are no other animals, creatures that get Huntington's disease except human beings, and I'm happy to talk about why I think some of that is, but be that as it may, you can't expect that. What you can hope to find is target engagement, some biological activity just to confirm your mechanism of action, things like that. But if you really want to understand translation and how it might work in patients, you have to be in the humans. Now, it happens that there are some drugs that, only work in humans or primates. An example of that is the pain molecule out of Vertex. I'm not paid by them, just a disclaimer. I don't work for them anymore, and I don't get paid by them. I don't even have stock. And but the point there is that those molecules only worked in humans and primates, and so we had to find a way to get human DRGs to try to ask whether our compounds actually worked. So you might say, yeah, but the FDA is never going to approve this if you don't have an animal model. Wrong. That wasn't true for the pain, and for cystic fibrosis, there was no even vaguely decent model for cystic fibrosis. And so the use of these primary human bronchial epithelial cells turned out even better than you might imagine, but that system was used to understand which compounds affected which mutations and where we thought those drugs might be useful. And so I think there's a lot of room for creativity on that side. But you have to not expect that your organoids or spheroids in the dish also are going to look like Huntington's disease. There will have features of Huntington's disease. But the other emphasis is, as Christian said, you have to look at a number of patients. You have to see what the commonalities are, and across the patient donors, because of course, just as no two people are cloned, those cells are not cloned either. And so there's just a lot of considerations. And so it is a little harder, it is a little more expensive, but in many respects, you actually save money because you have a chance to find something that will can translate into the clinic.
Your response triggered sort of a follow-up for me, if I may, around, if the target doesn't exist in mice, and you said you then used a human cell model and got past the FDA into the clinic without having an efficacy model, what was the thinking behind how to demonstrate safety if you don't see that target engagement?
That's a really good question, because a lot of people also say, well, if you don't know the target, how are you going to do toxicology? But you do toxicology the same. Knowing the target actually doesn't change your standard toxicology packages. Could there possibly be concerns? Yes, possibly. But I think that's true. Even when you think you know the target of your drug, I will tell you that most of the drugs that are on the market, you probably appreciate this, we think we know the target, but for a bunch of them, we don't probably actually know the target. And so you run your toxicology studies as you would.
Dan, same question for you. Yeah.
Thanks. So I actually want to acknowledge Vertex as well. I started my career at Vertex, but it's been a long time since I was paid by Vertex. But two things. One, when we were earlier in the stage of QurAlis, we would point out that Vertex had actually gone to the market without an animal model and gotten approved drugs when people were skeptical about that. I'll also point out that Peter Grudenhouse, I don't know if you knew him.
Very well.
Peter was one of the lead chemists on the cystic fibrosis project, and he once said facetiously that he was asked about animal models, and he said he hoped that someone would develop one soon because it would set the competition back by years. So, I think that one of the things that we have to think about human systems as we go forward is that we do have, we have neurons in a dish, and maybe if we're a little more sophisticated, we have neurons in a dish sitting on glia, and that isn't actually a brain, and so as we get to organoids, we get a little bit more, more representative cell types, and I think that as we advance, like getting those models set up and running, and figuring out what we are going to measure and how we are going to measure that is really important. I will say one other thing about the genetics, and everybody likes a genetic disease, monogenic disease. Monogenic diseases aren't always 100% penetrant, and so there's more going on than what that one monogenic factor is. And so whether that's other genetic factors that are suppressors or enhancers, or it's some sort of environmental trigger that triggers some change, we don't know that. So I think that that's an important component. And it's quite clear if you look at, the military has a lot of issues with continuous trauma, essentially, low-level trauma. Those individuals don't necessarily have what we would consider, genetic risk, and yet they get these neurological diseases. So there's more to it than just your genes.
Great, thanks. So this morning we talked a little bit about biomarkers that came up during the whole day, and also digital endpoints that are being used more and more. And I think the scientific community certainly sees the value in monitoring patients 24/7 versus a sort of a diary or some ad hoc patient visit. So going back to you, Beth, how do you use a biomarker strategy and a digital biomarker strategy or digital endpoint strategy in your development program?
Yeah, so we're not quite in the clinic yet, but we pay a lot of attention to how we might run our phase 1. And I think I'm a huge proponent of both, obviously, blood biomarkers, but a lot of the digital biomarkers, I think, are critically important. So, pardon me. So I think what we don't do enough of, we don't collect enough data. And I know everybody's probably drowning in data, right? But let me clarify what I mean. And that is, we need to have more frequent readouts from patients than the quarterly visits or intermittent visits. And I think because, let's face it, the patient comes in and they want to do the best they possibly can. And if they're on a clinical trial, they want to tell you something good has happened, right? I mean, that's just human nature. And so what you need to really see is, we all understand, and if you think about your grandparents or anybody you know who has a chronic illness, there are good days And there are some bad days, and there's everything in between. And so you really need to monitor what the sort of overall average of that is. And that's why I think a lot of these digital markers, but there's also, EEG and a number of other factors that can be monitored from home and uploaded via the cloud, et cetera, and really collect, get a good picture of what's going on for these different patients. And I think when we really embrace that and actually have a lot of that data, and I know that's happening, and it's beginning to happen, and everybody's thinking about it and talking about it, but I think what we'll see looking back on that is you'll see how, patterns of response and patterns of non-response. So the other piece that I always think is important to plan for are how are you going to look at responders and non-responders, because that's another huge wealth of data that helps us understand. Because remember that, when I talk to HD patients and their families, everybody there is hoping that one trial is it. Mm-hmm. That's it. We found the magic bullet. We're done. But I think what we all understand is that, and what we've heard today, is each trial builds on the next trial, and you incorporate data and try to understand. And so that's why I think, we really need to embrace and use much more of the genetics, the epigenetics, and any modality that you can really envision, in addition to the traditional blood biomarkers, to understand what's going on.
Can I get in on the blood biomarker thing?
It's a panel, yes.
Yeah, because we've heard a lot of interest in this, and maybe I'll just rain on everyone's parade for just a minute, because I'm a scientist, and we tend to like to do that from time to time. Sure, blood-based biomarkers, very convenient, easy to collect, but what for? I think if your cell type is CNS specific, your target cell type, sure, maybe what you're measuring in the periphery can be reflected in terms of the biomarker in the periphery can be reflective of something. But if you have a background of, if your cell type is, let's say, microglia, which is, even in the brain is about, 10% of the cells. And if you say that, if you acknowledge that microglia are macrophage-like, they're infinitesimally small, representative of, and you know this very well, of the total macrophage population, which is what will dominate your, the immune background immune signature in your serum or plasma. So I think, in some cases, if you're looking for, evidence that you've modulated your cell type in the compartment that you want, which is often very important from that key translational piece from phase 1 before, and building this interest, whether or not you have something that have established directionality of pharmacology or function before you can build interest, whether it's from investors or what have you, or your own conviction, to do a phase 2, which will be a big leap of faith and require lots and lots of money. I think you can, if you focus just on serum for certain types of biomarkers, you'll be subject to, false negative, or in some sense, false positive. And so I think that's maybe a little bit of a specific issue where if the cell type of interest is highly represented in the periphery too.
So I'll push back a little bit. I think that, it is important to have biomarkers, and working in ALS is particularly with intrathecally delivered ASOs, you get access to CSF, but even, figuring out what's going on in the blood is important. And, I don't know how fast progressing. Some neurological diseases are fast progressing, but a lot of them are quite slow. And so if you want to look at at whether you've engaged the target. If you can't get to the compartment, if you can't engage the target, and you don't have some sense that you engage the target, then you can be committing yourself to a very long trial, not really knowing. In ALS, for those of you who haven't followed it, there's actually now evidence of restoration of function in SOD1 patients from the tofersen, who are on tofersen. There's a FUS trial that's ongoing. There's a video, it's probably on YouTube, a patient named Anna. Started out, could barely walk upstairs when she went on the therapy, was on a portable respirator. Last year, she attended the M&D meeting last year, and she can now walk downstairs unassisted without ventilation. So this is really a massive amount of restoration of function. Whether that's going to be true of all patients, but if you don't know that you're modulating the target, are you really going to run the trial for 2.5 years to figure out whether or not you're restoring function?
Well, I don't think I was, sorry, just to qualify, advocating blindly going forward. What I was saying is there's a replacement for seeing target engagement in the CNS, which I would say if you have a choice, you would, and you can enroll and get CSF, you would do that. And I probably would agree with that. Rather than say, under no circumstances would I would say that you could just fly blind. I think in the earliest session of we heard that is now an issue of the past, that we're doing a better job of establishing clinical pharmacology. Yeah. I wouldn't want to turn back the clock.
The investors are starting to come in for the next panel, so you don't want to look like you're just going to be spending their money.
It's also, those discussions keep the panels memorable, so it's all good. But speaking about the clinical development path, I think Vigil, you guys have really embraced the natural history study of ALSP, a disease that we don't know, at least I don't know that much about. And tell me what was the thinking behind really putting an emphasis on that and studying that disease as it naturally progresses to help your clinical development?
Yeah, that's, I would say, one of the proudest things I've been part of at Vigil Neuroscience. And obviously, we work really hard as drug hunters and bringing forward therapeutics. And I'm proud we brought 2 molecules in just 3 years into clinical interrogation, one for that patient population. But these non-interventional trials are a two-way gift. One, you're studying something systematically about the disease, and you are, laying the framework for not just your own phase 2 study, but for those who may follow. And you're doing that up to a rigorous standard with translation in mind, and that is why we studied this. We started an Illuminate trial for ALSP. So just briefly, ALSP, it's, in terms of rare disease, there's, it's not entirely known how many patients there are, because this can be tricky, but on the order of, say, 15,000 patients in the US. We think there are more because, as we talked about earlier in the session, that genetic testing is not a routine part of all the workup of all neurodegenerative diseases. And so for non-circuit-based diseases, such as ALSP, which is a microgliopathy, it may manifest and look like a bunch of other diseases. So we believe that there are probably hidden patients. So an Illuminate trial really starts to systematically say what the disease looks like and what the biomarker changes are. Some of them could be very important in 2 different ways. One is you are maybe identifying biomarkers that are unique to that patient population, to that genetic dysfunction. And so maybe you can go with that and try and round up the patients that are lost in other types of clinics because you have something unique. So we've done that in that case. And another important one is as you your non-intervention study is often for biotechs a feeder for your phase 2 study. You're starting to get these biomarkers, and that allows you to make correlations between clinical scales and movement on whether they be serum or CSF or radiological biomarkers. And if you can establish that correlation and that surrogacy, you can now go forward and actually have regulatory discussions around accelerated approval. So, and the FDA is, receptive to these discussions. We've been happy to have those interactions. So I think that is another important piece, is paving the regulatory path forward. The third, of which all just occurred to me as we were talking, is maybe around reminding the patients that there are people out there that are interested in their disease. And a rare disease standpoint, this is really important. That you're working on their disease really matters, and building up a strong relationship with advocacy groups can help your future recruitment. So that's also important, and establishing that trust with them is so that they will follow through with your study, and that they will do, say, CSF collection if it's important, or they will commit to a genetic test across their family, which is not always easy to convince people to do, to increase, the number of patients that you can study the disease. So I think there are many knock-on benefits of doing these non-intervention studies. Yeah.
I want to add one thing that, Beth, I think you mentioned the amount of data we have, and that so one of the things that we find is that as the field advances, what we have is a wealth of data, but we haven't collected the newest data on those patients. So there are patient registries. I was talking to one recently. They have data on 23,000 ALS patients that they've collected. I mean, that's an enormous number of ALS patients. A lot of it is 10 or 12 years old, and so they have what was collected 10 years ago and 8 years ago and 5 years ago as far as what was the leading thinking about the disease at that point. But not everything that was thought of as important 10 years ago is what's thought of as important now. And so when you get down to it, of these 23,000 patients, only about 160 of them actually have the data that you're looking for, right? And so now you're actually looking at 160, not 23,000. So we have a lot of data, and it's not always the data that we actually want today.
Great point.
Great points. I'm getting the subtle cue here and I had it on my list to look at the audience. I think anybody that has a question for the panelists, if you please come up to the microphone.
So one of the topics you touched on is the importance of human models, but most of you were talking about organoids, in vitro models, and Dan mentioned there's some diseases for which there's thousands of patients who've been profiled in various ways, biomarkers, et cetera. Are you guys using any of that human data to think about to validate your targets, to think about your clinical trials?
I'll jump in there. So first and foremost, before we start working on targeting or what we're going to hope to measure in patients, we look at human genetics. So there are lots of clues from human genetics. In the case of, say, Huntington's disease, if you have only one intact gene, so basically one allele, those folks are fine. That suggests 50% activity is okay. That gives you some guidepost. There are cases where you don't have as much expression off the mutant allele. Those patients actually have later onset, or you have a mutant allele that is more highly expressed, and those patients have earlier onset. So we use those cues to help us understand what we think we understand and what we want to go after. So certainly genetics, and certainly I think what I'm very proud of the HD field for, and I had really, that's not my doing, is the way they've come together, taken the tominersen trial that, didn't turn out as well as everybody hoped, and use that data and a lot of other data from natural history. There are a number of very long-term natural history studies in Huntington's disease. They maybe haven't measured everything, but they've measured a lot of different things and tried to put all that together to come up with this HD international staging system, which basically is a sort of platform by which, it's really still a research platform. Is this right? But here's what we measure at this early stage, like caudate volume, putamen volume. Here's what we measure at this stage. And I think those are the kinds of things that help you understand who to recruit, what to look at, and when. And obviously, we're always trying to figure out what else to look at. So definitely, I think the patient information is huge, especially the natural history. And even if you haven't measured everything, at least you have some idea. Because in a lot of these cases, as I think everybody's heard already today from almost everybody, the disease, these neurodegenerative diseases do not progress identically for everybody. And in a lot of cases, they don't even present with initial symptoms that are identical across individuals, even if they all have the same mutation. And so all of this is important to be able to take into consideration.
So we actually have a collaboration with a company called Unlearn. They're an AI company.
Yep.
They do digital twins. So the idea is to take the data that you have on your patient population and to come up with a digital twin for your population. Of course, you can collect more data today than some of the data that they have access to historically. And then you can have some level of, the idea is to get some level of prediction of what to expect if those individuals went untreated. To hopefully, lower your placebo group size. So.
Yeah, they're good. We were talking to them too.
Any other questions from the audience? If we don't, I'll probably task the panel with one final question. And that was a request by the panelists, so don't blame me for putting them on the hot seat for asking what breakthroughs can we realistically expect to see in the next 5 to 10 years in Alzheimer's and neurodegeneration.
Can I take out the word realistically?
Yes, yeah.
Okay.
Yeah.
I don't know. I don't think that was my word.
I agree.
I mean, the thing that I would really like to see is the ability to get a sense of what's going on in these diseases during life, and we're getting better at it. But, I once had a lawyer in a discussion say that, yes, we could biopsy the brain as long as we put it in the informed consent and the IRB was amenable to actually leaving it there. That said, if we could get differential gene expression in living patients across the various cell types of the brain in the various areas of the brain, that I think would be a huge breakthrough. I'm not sure that's realistic in, whatever time frame we're talking about. But.
I guess looking forward, I think about a couple things. One is I think that, the sort of genetic medicines have a lot of promise, things like ASOs and the antibodies, of course. But I see that to really have something that's hugely transformational, we will eventually be leaning on small molecules. Happy to have an argument with anybody. I am being a little bit controversial, sorry, provocative, I guess. But I think that we need to take one bite at a time out of neurodegeneration, and I think that now I think all three of us are sort of focusing on the genetic forms of the disease, but I wanted to comment that I think what we will learn from going after the different mechanisms and the different neurodegenerative diseases caused by particular mutations is we're going to learn a ton. And I think that that will help us address the forms of disease that currently we don't recognize as genetic. And so that's what I'm looking towards. For me, I think we need some really big successes, and I think that will come from the monogenic forms of disease, but I think that we'll have the opportunity to expand that much more broadly moving forward.
On my side, I would look forward to whether it's realistic or not. We'll see. I guess a few things. I think we've spoken a lot about genetics and what validation and confidence building that gives us. I'm excited to see this emerging field on the human genetics of resilience or healthy aging. I think we study a lot of risk, and often that points us to the antecedents of the disease, the causal seeds of the disease. I'm very interested to see how some of the genetics or omics around resilience emerges. I think that's pretty realistic, how deep it will go. Maybe we're having people live to 100 years old more often now, so I'm cautiously optimistic. I think a related one around that is, again, it's the question around the genetics of risk is we've talked a lot about biomarkers and being able to measure serum biomarkers longitudinally. I'm wondering if the genetics of disease progression is going to map out the same way as the genetics of risk, and I doubt it, is my sense. So if you can look at the trajectory or the slope of a disease and understand, what are the causative or what are the genetic modifiers of the steepness of disease progression, I think that could open up inspire a new set of drug targets where today we're focused on risk. Obviously, there are efforts going on in that space, so I think it's realistic. Dr. Tsai mentioned, the Swedish biofinder efforts. There are other ones, and the ROSMAP study's been going on for a long time, thinking about resilience. So I think these are really taking shape in a systematic effort. So I hope, right now I'm focused currently on risk genes, but I am really looking forward to expanding the repertoire of therapeutics in that direction as it emerges.
Great.
Thank you so much for ending it with a high note. And a round of applause to the startup heroes.